Hello, cell biology fans. Well, I'm sitting here at home and I'm babysitting the dog that had surgery this past week, and I thought I would give you the rest of the lecture on biosynthetic molecules and what happens after the generation of either soluble or transmembrane proteins. In our last lecture, we had talked about co-translational import through Sec61 into the ER, and what happens if you have an N-terminal signal sequence versus internal signal sequences. And then the next question really is, okay, so we've generated proteins. Some of them are soluble and they're in the lumen of the ER. Some are transmembrane proteins, and what happens next to those molecules? So one of the main features of both the ER and the GOLGI is the process of glycosylation. And glycosylation is the addition of sugars to newly made either lipids or to proteins. In this case right here, this slide is talking about the addition of sugars to newly made proteins. The picture shows that there are two steps, either in the ER or the GOLGI. And in the ER, one interesting thing is that we start the addition of sugars to proteins in the ER by adding a huge block of sugars. And as you see, it says asparagine. And what's going to happen is we call this N-linked glycosylation, because asparagine is an amino acid and its one-letter symbol is N. So in the process of glycosylation, it starts in the ER and it starts by N-linked glycosylation. So as the protein is being added into the ER via the Sec61 translocon, if there is a sequence of amino acids that is recognized for glycosylation, a block of sugars can be added to an asparagine. There's additional glycosylation events that occur following this. This is blowing this up for you a little bit, but you can see that on the left-hand side we have a block of sugars, so all of those are 6-carbon sugars that are added, and you see 2 yellows, 1, 2, 3, 4, 5, 6, 7, 8, 9 blues, 3 greens. So 9 and 3 and 2 is 9, and 2 is 11, and 3 is 15. So we have 15 sugars that are added in a whole block. These are covalently linked to one another, and they're covalently added to asparagine. Now in the ER, some of those sugars are actually now removed. You might ask, why do we add and then we remove? There's a lot of why that we can't answer. Whatever the reason is that we added all of these and then we take off some of the glucoses, it works. Remember, so processes that have been going on through evolution that work, we continue to use. So in step 1, we've added and then we started removing some of the glucoses. In step 2, you see we modify where some of the mannose residues are, so there's a monosidase. You don't have to know these glucosidase, these monosidase, the N-acetylglucosamine transferase. What I want you to understand is that the addition of sugars and the modification of those sugars results in different modifications to different proteins. Okay? You'll see that we add molecules that are called N-acetylglucosamine, N-acetylneuramic acid. They're called glucnec and NANA. There's sialic acid that can be added, there's galactose that can be added, there's mannose that can be added, and there's glucose that can be added. This is showing you that the sugars, that block of sugars that we add to the asparagine on the growing polypeptide chain as it's being placed into the ER, into the lumen of the ER, is on a molecule called dolicol. Dolicol has 2 phosphate residues and the sugars linked to it, and the addition of that block of sugars to asparagine occurs in the ER, and it happens because the dolichol is right next to it in the ER membrane. For whatever reason, dolichol comes up on standardized tests. MCATs, I don't know about PCATs, but definitely MCATs, dolichol is the molecule that holds the block of sugars that's added in the ER. So we call this glycosylation. You have glycosylation that occurs in the ER. You have processing of those molecules that are occurring, and this is called N-linked glycosylation. Once we do that N-linked glycosylation, we then start trimming off some of the sugars to make specific modifications. What happens if you have proteins that are being made in the ER, right? And some of them don't get folded properly. One interesting thing about the production of these proteins, is that proteins that are being made and co-translationally imported into the ER are being folded immediately upon being made, and they're folding to reach their lowest energy state. Every protein, every being wants to be in its lowest energy state. Remember, I told you, you go home, you're not at home for more than 5 minutes before you're sitting on the couch. We want to be at our lowest energy state. Proteins want to do this as well. And there are particular proteins called chaperones that help to guide the folding of proteins. Two of these are called calnexin and calreticulin. And you're going to see throughout your biology studies there are a lot of proteins that are called cal something. And what's interesting about that is many of those they function because they're in the presence of calcium. So just keep that in the back of your mind. If you see a protein with the name calnexin, calreticulin, calcineurin, they all function in the presence of calcium. And these act as chaperones, and even in the presence of chaperones to help proteins not to misfold, we still have a lot of misfolded proteins. Up to 80 to 90% of some proteins don't get made correctly, and they can really cause a problem. If they pile up in your ER, they will stop co-translational import. They will just gunk up the system. And we call this, quality control. So quality control is important, and we have to get rid of those misfolded proteins. Well, how do you get rid of a misfolded protein? Misfolded proteins can be exported out of the ER, and they're then degraded they're put back into the cytosol, degraded into amino acids, and those amino acids can be recycled. Another interesting thing that can happen is sometimes proteins can make it to maybe the Golgi, and they shouldn't have been there. And in this slide, I don't particularly care for the image because they're showing you a soluble ER resident protein. And they're saying it's the pink thing with a red head. And it looks like they're two separate molecules, but they're not. That is a sequence of amino acids that is folded into its tertiary or quaternary structure. You can't tell from this picture. So it is just a sequence of amino acids, and within that sequence of amino acids, it has a sequence of amino acids K, D, E, L, lysine, aspartic acid, glutamic acid, leucine, or KKXX, which is, what's K? So K is lysine, right? Ooh, losing my mind here. Lysine, any amino acid. And what happens is if proteins that are supposed to be in the ER, they have a KDEL sequence. We call it a KDEL sequence. That KDEL sequence is a retrieval signal. So we have ways of getting things that escape by accident. Let's say they ended up in a budding vesicle, leaving the ER, headed towards the Golgi, and they weren't supposed to go, but we need them back in the ER. These KDEL containing proteins can be recognized by a KDEL receptor. They're showing that to you here in green. The KDEL receptor recognizes the sequence of amino acids. It binds to it, causes it to be concentrated into a vesicle, in this case a COP1 coated vesicle that buds out of the Golgi and goes back to the ER. So we call this retention signals. This is a Golgi, right? I mean, an ER retention signal. KDEL is a sequence of amino acids. Remember what Harut's answer was the other day, a sequence of amino acids, and all that is something that can be bound by another protein. In this case, it's going to be bound by the KDEL receptor. That leads to budding from the Golgi, transport and fusion back to the ER. So there's two ways we can get things, right? So you have misfolded proteins that are going to have to get out of the ER, and they're going to get out of the ER and get degraded in the cytosol. You have proteins that get to the wrong place, and they can be retrieved through their KDEL sequences. And then what other things get made that are really important as far as ER goes? We talked about rough ER. Rough ER is where all the ribosomes come and do co-translational import via Sec61, but we also do membrane biosynthesis, meaning we make phospholipids, and that occurs on the smooth ER. I always thought this occurred inside the ER. It occurs on the cytosolic face of the ER. So the enzymes that are responsible for that are actually on the outside of the lipid bilayer for the smooth ER. So what happens is, as you can see in this image, you are making phospholipids, they're being added to the outside, and now you don't have enough on the inside compared to the outside. There's another enzyme called scramblase. And scramblase catalyzes that flipping of phospholipids into the inside, or it can catalyze it to the outside. And we talked about the unequal distribution of lipids, right, in membranes. Do you remember which particular, one in particular phospholipid we said is always on the cytosolic side of the membrane? That was phosphatidylserine. Phosphatidylserine was the only phospholipid that had a negative charge, and we talked about this in relationship to the plasma membrane. It's on the inside of the plasma membrane and gets flipped to the outside if and when there's damage to the cell. So in this case, remember, if it's on the cytosolic side, cytosol is cytosol, lumen is the same as being outside the cell. So if you have phosphatidylserine being made on the smooth ER, it's going to remain on the cytosolic face. The other phospholipids that are normally on the luminal side will be flipped in by the scramblase. Okay? All right, so the things that get made, proteins, lipids, these all are going to end up moving by vesicular transport. And let's talk about this transport from the ER to the Golgi. And it's going to occur through the ergic, ER Golgi intermediate compartment. Once again, I didn't make the names, so don't shoot the messenger, but it has another name, which is a little harder to remember, the vesicular tubular cluster. And I like that name because it actually is talking about the morphology of this organelle. It really is an organelle. It can be seen in the cell, and it has vesicles that bud off of it, and it looks tubule. And in this cartoon diagram, they're showing you ergic and VTC, and they're in between the ER and the cis Golgi. Okay, and so COP2 coated vesicles bud from the ER en route to the cis Golgi, and they bud using COP2. Now, once they bud, they have to fuse with the ergic or fuse with the VTC, and then they bud off of that and are then transported to the cis Golgi network. The Golgi, right, we've talked about the Golgi very little, but the Golgi is an organelle that is made of stacks of membrane-bound compartments. I think of it as a stack of pancakes. And it's not a stack of 1, it's not a stack of 2, it's not a stack of 3. Normally, it's a stack of 6. 6 pancakes. Why would you have 6 membrane-bound compartments that are all stacked together? And how would you keep that structure from not sort of just falling apart. How, on your plate, when you stack the pancakes together, they're being held together based on what? Gravity. Okay, what if there was no gravity? They would float away from each other, and that's what would happen in a cell. So what holds the Golgi together? We're going to look at that, and we're going to talk about that in a little more detail. So it's this stack of membrane structures, and they're called cisterna. Cisternae is the plural. And there's a cis, then there's several medial, and then there's a trans Golgi. Cis is closest to the ER, trans is furthest from the ER. Cis is where newly made proteins come from the ER, the ergic, and they are entered into the Golgi on the cis side. The trans Golgi is where clathrin-coated vesicles bud off, and the transport of newly made proteins go either to the plasma membrane or to an early endosome or to another organelle in the cell. The Golgi is actually made and it's sitting on a scaffold. So think of it as a mesh network that holds the Golgi together. And that mesh network is made of cytoskeletal components called microtubules. And how do we know this? Well, we can disrupt the microtubule networks using drugs, and we can see the Golgi actually just falls right apart. I love these pictures. I love EM. I love to be able to see these structures. And on the top left, I hope what you can see is this stack of membranes that are long and skinny, and that's your Golgi. And if you, on the top of that image is the nucleus, and between the nucleus and the Golgi, you see a whole bunch of vesicles, some other stuff that you can't really tell what it is. I hope you can see some rough ER in that picture. And that is showing you the rough ER, the ergic, and the Golgi. On the bottom left, you can really see this in very good detail. And if you count 1, 2, 3, 4, 5, 6 membranes, you say, ooh, it looks like there's 7. And what that is the cis Golgi and the ergic, or the VTC, are really hard to tell apart. You can see the rough ER, and you can see buds, and you can see vesicles in the bottom left picture. In the top right and the bottom right, those are immunofluorescence images of 2 different distinct kinds of cells, and they're both staining for Golgi compartments. In the top, you're looking at, I think it's some sort of mammalian cell, and the Golgi is really sitting very close to the nucleus. You don't have Golgi all over the cell. So we say that the Golgi is perinuclear. In the bottom right-hand picture, that is a picture of a plant cell, and it's very different. I hope you can see the difference is that you have Golgi all over the place. There's Golgi everywhere in this cell. So different cells will have different morphologies. In general, I think about mammalian cells, I think of the Golgi as being perinuclear. Lots of things end up being perinuclear in the cell. So just remember, we're talking about the Golgi being stacks of membrane called cisterna. And why would you have different membrane-bound compartments? Because there's different proteins in each one, and they do different functions. So they're going to have different enzymes, and the primary goal of the Golgi is modification of proteins, and that modification is going to lead to different functions of proteins, and you do it with different enzymes. Back to the glycosylation. So on the left, it shows you the beginning of the glycosylation occurs via dolichol in the ER. We add N-linked sugars to asparagines, and once that molecule buds out of the ER, goes to the ergic, then goes from the ergic to the Golgi, we see that there are additional enzymes, right? In the Golgi, there's the Golgi monosidase, there's the N-acetylglucosamine transferase, there's the Golgi monosidase 2. You don't have to remember those names. There are different enzymes in different compartments that are going to modify the proteins. It's going to modify the proteins by changing the carbohydrates that are added to them. But there are also additional modifications that can occur. So there's glycosylation, and the N-linked glycosylation begins in the ER. There's also O-linked glycosylation, and this is a little bit of a trap, and I'm not setting a trap for you to fall into, but the N-linked glycosylation is adding to N, which is asparagine. O-linked glycosylation, think, are there any O? No. What is O? What is O-linked? O-linked, I mentioned to you when we were talking about specific amino acids. Specific amino acids get glycosylated. Two of them are serine and threonine. Okay, they both have hydroxyl residues. So the hydroxyl residues allow for the addition of sugars. So those hydroxyls in serine and threonine get modified by O-linked glycosylation. Was there another one that had a hydroxyl? Do you remember? Tyrosine. What's interesting is tyrosine doesn't get glycosylated nearly as often as it gets modified by another mechanism, and that's going to be phosphorylation. So if you're thinking of glycosylation, think of N-linked glycosylation on asparagines and O-linked glycosylation via serines and threonines. But those are just sugars. We can add sugars. You can also phosphorylate proteins. You can add sulfur groups. You can do all sorts of modifications. You're going to see there are modifications that lead to different changes in different functions. And on the right, the picture is showing you that even though they're showing you only 5 cisterna of the Golgi, that each of the different cisterna has different enzymes in it, and that leads to different modifications leading to different functions. We didn't really talk about budding from the ER. The budding from the ER is regulated by coats, rabs, and snares. Interestingly, it's all variations on a theme. Remember, we said the coat that comes from the ER to the ergic is COP2. There is a GTPase that plays a role, and it's called SAR. And SAR is the GTPase. COP2, remember, was Sec 13 and 31 and Sec 23 and 24. And in the picture on the top left, they're showing you cargo that's bound to cargo receptors, gets recruited into a budding vesicle, and the budding vesicle is regulated by COP2. COP2 causes curvature of the membrane, concentration of the cargo, and once it buds off, we have uncoating, and that goes to the ergic, fuses with the ergic, etc., etc., etc. Different coats, different GTPases, all of this leads to membrane transport, vesicular transport. Here's a more detailed picture of that. You see SAR1 in its GTP-bound form is bound to the membrane, right? When it's bound to the membrane, it recruits the coat. The coat is Sec 23, 24. The addition of Sec 23, 24 results in the recruitment of Sec 13 and 31. All of this leads to the concentration of cargo. That all ends up in resulting in the formation of the COP2 coated vesicle. So COP2, think of COP2 as forward transport. Forward transport is called anterograde transport. That's COP2. COP1 only does retrograde transport. Retrograde transport, and we know that COP1, functions in every stack of the Golgi. So you have COP1 dependent transport from the trans Golgi to the cis Golgi to the medial Golgi to the cis Golgi. I'm sorry, let me start again. You can have COP1 retrograde transport from the trans Golgi to the medial Golgi to the cis Golgi back to the VTC or the ergic back to the ER. And we can actually tell this because we can isolate COP1 coated vesicles from each of those places. Here's the ER picture once again. It looks no different than the COP2 coated vesicle. You can see the membrane. The white part on the EM is the membrane, and all of the outer portion of the dark is the COP1 coat. I didn't mention what COP1 is made of. It's made of a protein complex, macromolecular complex, just like COP2. Don't worry about the names of the proteins. here this is showing you the retrograde transport, and it also is regulated by a GTPase. The GTPase in this case is called ARF. ARF is bound to the membrane in its GTP bound form. It recruits additional coat complex molecules. That leads to the concentration of cargo that's going to go backwards, and that results in the formation of a COP1 coated vesicle concentrating cargo, causing curvature, leading to budding and transport and fusion. Remember, uncoating occurs really rapidly. No matter what, a vesicle cannot fuse unless it removes its coat. And we didn't talk specifically about the molecules that help in uncoating. I said it requires energy, so it requires ATP, and it requires another molecule called NSF, which is N-ethylmalamide sensitive factor. So NSF is found in cells, and if you add a lot of NSF, it leads to the uncoating of vesicles really quickly. That leads to fusion. All right, that ends the lecture on exocytic transport. The only thing I have left for you is I provided you with this little sheet, and I thought what you could do is, if each of these was a signal sequence, you could look at each one of these proteins and you could start to try to understand what would the topology of each of these proteins be. The protein on the top has 4 signal sequences. The protein in the middle has a lot, right? It has an N-terminal signal sequence, 1, 2, 3, 4, 5, 6, 7 internal signal sequences. What would that protein, what would each of these proteins look like if it was on the plasma membrane? Where would the N-terminus be? Would it be on the extracellular side, or would it be on the cytosolic side? And what would the hydropathy plot for each of these proteins be? Okay, where would glycosylated amino acids be? And one interesting thing about that is, if you think about where glycosylation occurs, right? Glycosylation occurs inside the ER on asparagine residues. So that occurs on residues that are luminal, all right? When that protein gets transported up to the Golgi, the only amino acids that can get modified are the amino acids that are sticking into the Golgi lumen. And so, which portions of these proteins could have glycosylated residues? These are the kinds of questions that I would ask you on an exam. So we'll work on this in our next in-class lecture. We'll talk more. I'll give you more examples of the kinds of questions that you should be able to think about and answer. You should be able to apply the concepts that we've talked about and go over this in class the next time.